So this meeting is an historic event. You know, all know me. I'm Victoria Sork, and I certainly never thought that I would be having a salon with the Dean Circle and board members and other special friends of life sciences virtually. I decided that it's only fitting that I greet you to our salon by sitting in the courtyard outside of Hershey Hall and near the Terasaki building, in case you were wondering where I am. And I'm also really overwhelmed because we have incredible participation. Typically, when we have a salon and you have to drive to campus, we're happy when about half of everybody in our groups is able to come. And today we have over 50 participants, which is about twice the number that I thought we'd have show up. So I'm excited that you're here. In many ways, what you're going to experience is kind of what our students and faculty are experiencing, what it's like to teach remotely. And so like we do in all of our salons, we always like to introduce you to our amazing faculty and their ongoing research. And it seemed really appropriate that we have a salon tonight that's on the theme of COVID-19. And in particular, what I wanted to do was feature our faculty who have expertise with different aspects of what this disease is causing and its causes and what it means to society. So I've invited 3 of our faculty to speak. And the first one is engaged in many studies that are being widely cited by the New York Times and all sorts of local media. And I'll introduce him in just a second. And then 2 other speakers who are going to be panelists talking about different aspects. So let me get started by first again thanking you for coming. And now I'd like to introduce the first speaker. And the first speaker is going to be Jamie Lloyd-Smith. He's a professor in Ecology and Evolutionary Biology and Life Sciences, and he's also a professor in the new Computational Medicine program. Jamie has been exploring the ecological and evolutionary dynamics of infectious disease in both animal and human populations, with an emphasis on the emergence of novel pathogens, which certainly this COVID-19 is. His group combines mathematical models, statistical analysis, and laboratory, clinical, and field studies to study infectious disease, and he's done work on influenza, monkeypox, leptospirosis, and now COVID-19. So with that, I think I will pass the mic and the video over to Jamie Lloyd-Smith. And let me say one thing. If you'd like to ask questions and you didn't read that preview, there's a little Q&A button on your screen at the bottom. Either during his talk or at any point, you can just type your question. And what I'm going to do is monitor those questions, some of which we'll answer right after Jamie finishes speaking. And some of them I might save for the end. And I will also save and introduce our other 2 panelists after Jamie's done. So now that I've given you instructions on how to ask questions, you can ask them at any time, but they won't be answered until he's done with his talk. And I'll kind of go through and screen them. Hopefully we'll get through as many questions as possible. So now let me introduce Jamie Lloyd-Smith to talk about his research on COVID-19 spread and infection. Go ahead.
Hello, good evening. It's a pleasure to have the chance to speak with you about the work that we've been doing on COVID-19. With apologies, I'm going to be showing you some slides. And so I'm sharing my screen now, and I will try not to make it too boring, despite it being a PowerPoint presentation. So yes, I'm going to speak with you about COVID-19, how it transmits, and how we can stop it. Now, the long-term goal of my group's research program, as Dean Sork said, is really to put together knowledge across scales and across species boundaries to both understand and work to prevent the emergence of new pathogens. So when I say scales, the little diagram at the bottom shows that there really are important processes that act right down to the molecular level, DNA, things that happen in cells, in tissues, up to the dynamics of what occurs inside an individual patient, and then to the population level spread processes. So that's crossing many physical scales. Across species boundaries, we know that most of our new pathogens as humans come to us from other animals. And so this connects epidemiology and clinical medicine to ecology and studying how diseases work in wildlife populations. And so the schematic at the top shows that there are transmission dynamics inside animal reservoirs. There's a spillover event when the virus or bacterium comes over into humans. And then there's either a greater or lesser degree of spread between humans. There may be evolution that occurs during all that process. So many fascinating processes come together. It's actually a quite complex field, putting together all these ideas. But the strategy that we take in my group is to basically iterate between mathematical and computational models, which we use to put these ideas together in a formal framework, and then data that come to us from either clinical settings, laboratories, or the field. And by going back and forth between these, we hope to generate new insights. So today I'm going to tell you about work we've been doing on COVID-19. And there are three big questions that we've been weighing in on. The first is who is transmitting and how can we find them? The next big one is how does transmission work, and hence how can we stop it? And then in ongoing work, we're thinking about what's going to happen in the coming months and years. So first I want to talk about how to find infected people. And this is work we did back in January, right when the pandemic was not a pandemic yet, looking at how effective symptom and risk screening, the kind of thing they do in airports, is at detecting COVID-19 infected people. And in the schematic at the bottom, you can see there may be screens that occur at departure. Further biology, in terms of showing up with symptoms, can occur during the travel time of an intercontinental flight. And then there may be another screen that happens at the arrival end. So we began this work thinking very much about the travel context, but as we did it, we realized that it's actually relevant in many other settings as well. This type of symptom screening is used to determine whether people are safe to come to work, whether they can be the ones preparing your takeout food, and so on. It's especially important in the unfortunate setting we find ourselves in, where there's not enough access to the more rigorous molecular tests. So what we did is we built a mathematical model that basically put all the factors together that determine whether this type of screening will work. And these are things like the incubation period, that is the time from when you get infected to when you actually begin to show symptoms. Also the proportion of infected people who actually end up showing symptoms, which is a very hot topic in the COVID realm. And then the proportion of the population who may know they've been in a risky situation, whether it means they've traveled from a hotspot or they actually were sharing a living space with a known case, for instance. So this model captured the knowledge and all the uncertainty about COVID-19 as of late January. And then I've already shown you the first take-home result. And there you see the three little bars represent screening being applied at departure or arrival or at both of those points. And the red colors, the big, unfortunately very large red bars, show the likelihood that an infected traveler is not detected by any of these modes of screening. And the little blue bars show the likelihood that a case is detected. So you can see right away that this is not a very reliable approach, unfortunately. And this is actually the relatively best case scenario, because this is the situation where only 5% of cases don't show strong symptoms. And there's been a lot of attention in the news about this point. It turns out that probably a much larger proportion of cases don't show symptoms. And we anticipated this back in January when we were writing this paper. So we also considered scenarios where either 25% or 50% of infected people never show the symptoms that would trigger the screening. And you can see the big red bars, which mean failure of the screening, just get bigger and bigger. So the take-home message from this is that this type of screening for symptoms and known risks will detect less than half of infected people in the very best case, and maybe as few as 1 in 10. So it's really not a viable standalone strategy to prevent importation of the disease or to really to screen people in occupational settings. And I think recent news about the early arrival of the virus into the United States really bears this out, unfortunately. Okay, moving on to the next big question. This is, how does this virus actually transmit? And hence, how can we stop it? And so I've been working with scientists at the National Institutes of Health who actually have the virus itself and the facilities to work with it. They're wearing the big moon suits and all that to keep themselves safe. And they've been doing experiments to determine how long the coronavirus can remain infectious in the environment. And this is crucial because it determines infection risk. We were initially focused on healthcare settings, but it turns out, of course, it determines risk in all kinds of settings. So our collaborators put the live virus onto various surfaces, and they also used a machine to spray it into a mist, into an aerosol, and then measured how long they could still detect live virus after doing this. And then my team modeled the data to extract findings in the most robust possible way. And this work was published about a month ago in the New England Journal of Medicine. Showing you the results here, these are the raw data at the top. And what it shows is for the aerosols on the left and then 4 different surfaces that we looked at, it shows basically how the amount of virus, that's the titer, changed through time as we resampled from these different surfaces or environments. And the red shows the data for the novel coronavirus. The blue shows the data for its close relative, the original SARS coronavirus from back in 2003. And the first take home is that the 2 viruses actually behave a lot the same. But then from those data that we see, and basically how long it takes for the little bars to get down to that dashed line, which represents the limit of what we can detect, there's some take home findings such as the live virus could be detected for at least 3 hours floating around in aerosols. And then for 4 hours on copper, 24 hours on cardboard, 72 hours on steel, and so on. But I have to emphasize, those numbers aren't sort of magic universal solutions. They really depend on how much of the virus you started with when you contaminate any of these surfaces. And I'll come to that point. I do want to remark, though, Dean Sork was describing some of the media attention. And I have to say, in my couple of decades as a working researcher, I've never experienced anything like the interest that we've had in this study. And in fact, I went Googling to find a nice picture to bring these slides to life a little bit, and found many images like this. This one's from the Boston Herald, but it's basically a graphical depiction of the results coming from our study. And there were lots of people asking about, how should I handle my groceries or my pets or you name it. So this certainly spoke to a very reasonable and prevalent interest in the broader public. But I want to make the point that these durations really are not absolute quantities, and instead it's better to think of this as a process. And so what my group did is we fit a statistical model to the data to basically describe what was going on to generate those numbers. So the number of viable viruses that are still infectious is decaying through time, almost like a radioactive substance. And so we fit these curves that are shown in the sort of clouds of blue and red lines to estimate that decay rate. And from that, we can estimate the half-life of the virus in each of these environments. So that's the duration of time it takes for the amount of virus to drop by a factor of 2. And this is a much more kind of universal description of how the virus operates. So here are the half-life estimates. Oh, we skipped ahead. Okay, I made a mistake in my animations. I apologize. At the bottom, there are little half-life estimates that show things like the half-life of the virus in aerosols, for instance, is about an hour. So that gives us a better sense, depending how much we start with, of how long the virus will remain viable and infectious in that environment. Now, this next slide shows there was actually a huge amount of interest in this finding about survival in aerosols, to the point where the World Health Organization, based on this study, altered its guidance about the risk of airborne precautions, in particular for medical staff, but there is ongoing discussion of whether airborne spread is an important risk in the community. And then much media interest as well. I want to point out there's that LA Times article, which maybe some of you saw. This is about a choir practice that happened up in Washington, where the people, they got together, decided to have a rehearsal. They were very careful, nobody touched, nobody hugged, they all used hand sanitizer. And yet 3/4 of the people, 45 people got infected in the span of a 2-hour rehearsal. And I helped the reporter with the story. He got back to me and told me that has broken all records for interest for the number of clicks for an LA Times story. So again, it really speaks to how focused the world is on this pathogen and on understanding how it transmits, because of course that determines the risk to every one of us. I should also say that I had a lot of contacts from medical personnel, so healthcare workers and healthcare administrators, who wanted to know how they can improve safety precautions based on what we had found. And in talking with them, I realized that this answer is really not sufficient. We have to think more deeply about what is involved in airborne transmission. So that's a study we're just trying to wrap up now, but I'll tell you what we've learned. So for an airborne infection event, you basically need 3 things to happen. You need some process to create aerosolized viruses. And this could be somebody coughing, sneezing, or even just breathing. But there are also certain medical procedures that are known to put viruses into the air in a dangerous manner. Then the virus needs to remain viable, infectious, and remain present in the air. And then some human, a susceptible human, needs to come in and be exposed by breathing that air and get infected. So there's a lot of science behind each one of those bubbles. But to make a long story short, we've built a mathematical model that integrates together what we do know. So the virus decay rate from the prior study I just showed you, also knowledge of aerosol physics, so how these little droplets float around in the air, and dose response theory, so what we understand about how a given exposure dose will give rise to infection. And we also use the model to work around the elements that we don't know, things like how fast do patients put this virus into the air in aerosol form, and also a crucial knowledge gap of what exactly is the infectious dose. In any case, we built this model to put together everything we know to provide evidence-based guidance to healthcare workers, because it was clear there was an acute need for that. And this is a complicated slide, but I'll talk you through it. At the top, these are sort of the take-home results from our modeling analysis. And what this shows is the probability that a person will get infected as a function of time since a contamination event. So at time 0, somebody does an intubation procedure and puts virus into the air, for instance, and then that decays over time. And then the different figure panels show increasing ventilation rates, going from a situation with no fresh air at all to something like what you'd see in your home, to a standard patient care hospital room, to an operating room or negative pressure room where they're really moving air through quickly. And the 3 different colors show 3 different scenarios of exposure, low, medium, or high. And the big take-home point here is that ventilation is overwhelmingly the most important factor here. So even once you've accounted for all the other elements, ventilation really dominates, which is an important take-home message for people in the field. But also we want to emphasize that even in the highest ventilation hospital settings, there can be significant risk that lasts for tens of minutes, up to 30 minutes or more, depending on the contamination level. So we're working further to calculate the waiting time that you would need to reduce the risk to a given level, and also to account for the fact that many healthcare workers are exposed multiple times in the course of their work, or for wearing masks. Now, I think I'm running low on time, having too much time telling you these stories. So I just want to quickly show you one last study that we just have in revision now. This is about the mask shortage. So masks, N95 masks, we've all been reading about how there aren't enough of them. And they're meant to be used only once, but healthcare workers are being forced to use them multiple times. So we tested 4 different methods for decontaminating them to kill the virus on them, and measured how fast those methods killed the virus, and also the impacts on the performance of the mask at filtering out aerosols. So here, this little plot summarizes the results. On the up and down axis, it shows how fast the virus is killed. So ethanol and VHP, that's vaporized hydrogen peroxide, very quickly kill the virus. And on the other axis, it shows how well the mask still works. And if you're on the right-hand side of that red line, that means the mask is still working well. So that looks very promising, but that's after just one round of decontamination. We tested how 2 and 3 rounds affect it. And you could see that, for instance, the yellow blob, which shows decontamination using alcohol, ethanol, it continues to kill the virus well, but it also kills the mask. Whereas the blue circle shows that vaporized hydrogen peroxide both kills the virus quickly and preserves the function of the mask. So we put that forward as an important option for healthcare workers in other settings where they're running short on this crucial protective equipment. Okay, I'll wrap it up at this point. I'll just say that we have several other studies ongoing on this important disease. So we're studying the virus's survival in various bodily fluids to learn about other modes of spread. We're looking at how that survival depends on different temperature and humidity conditions, which may be involved in driving seasonality of transmission. And we're also studying the dynamics of antibody levels within patients after they've been infected, both how the antibody levels go up and then how they decay away again. And this will be crucial for understanding the accuracy of antibody tests, which are going to be a key tool for how we approach reopening society to understand who's being exposed and who may be immune. With that said, I will show you this horrible picture of a sneeze. And if there's any time, I'll be happy to take some questions.
Thank you very, very much, Jamie. That was really a great overview of your research and how important it is to one aspect of the coronavirus. I'm going to remind everybody, I know most of you, there's a little chat, there's a little button there, just type in your questions. I have a few questions now, but there's, usually I have to encourage you to get started, and once I do, the questions just keep flowing and flowing. So I want to encourage everyone to type something in, but I'll give you some questions that are now here. One is, does the leaving masks alone for time decontaminate the mask if you just let it sit?
It does, but it does so much more slowly. So the experiment had a control treatment, which was basically no treatment at all. And it found that after something like a day, or possibly 2, and again, this will depend on contamination levels, the virus would be undetectable in the mask. But that's in comparison to a few minutes under these different decontamination methods. But for those of us who have masks we're using at home, if you just set them aside, especially if you put them in the sun, that is, if you leave it long enough, the virus won't last.
So just following up, can one sterilize a surgical mask by heat? If so, how much and how should it be applied?
Yes, you can. That was one of the treatments in this study. So in the study, we used, or I should say my collaborators who did the experiment, used 70 degrees Celsius heat. So it's actually about the temperature of a blanket warming oven in a hospital for 30 minutes. And that was quite effective at killing the virus. But over several applications of this heat, it did impair the mask performance. So the mask didn't pass its fit test as well. So it wasn't as good as a hydrogen peroxide in that way.
Great. Can viruses spread through the air conditioning in restaurants?
Good question. So this is something there is a lot of controversy about the potential for airborne spread in the community. The short answer is they certainly can in principle. And there is actually one example, I think from Hong Kong. There's a study where they have remarkable data of where the air conditioners were, where the airflow went, and a bunch of people who are sitting at different tables. And there's basically a line of tables where infection propagated from one table to another, and the people who are not sitting in the flow of the air conditioning did not get infected. So that's a pretty clear indicator that it can happen. And I would say it's a very rare sort of field result where it comes through so clearly. So unfortunately, yes, it is possible.
So just while we're talking about methods, what about microwaving?
That was not in the study, so I can't answer in any formal sense. You know, I don't know what an N95 would do in a microwave, because microwaves, as I understand them, work by kind of heating up the water in your food. And so if you have a dry mask you put in there, I'll throw that one open to the panel. I can't give a good answer, I'm afraid.
There's some other questions here, and I think I'm going to save them for the end because we're going to have other panelists talking, and they may also be able to address these questions. So if I didn't answer your question now, I will be holding it for later because there's still a few good questions that haven't been addressed. So knowing that Jamie's talk would only, his research, of course, doesn't raise every possible issue about COVID-19. So I asked two of our faculty who have related expertise if they would come and make a few remarks and then also be available for question and answer. So the next person who's going to be presenting is Professor Oliver Fregoso. He's an assistant professor in the Department of Microbiology, Immunology, and Molecular Genetics. And he received his PhD from Cold Spring Harbor Laboratory and did his postdoctoral work up in Seattle at the Fred Hutchinson Institute. So the reason we invited him is that his lab studies address the molecular mechanisms of viral replication. And his focus has been on HIV and related viruses. And if you look at the news, a lot of what we're looking at now about how things spread and what might be happening with the coronavirus have been based on what we learned from HIV. So his background here is very relevant. He's particularly interested in understanding how viruses usurp the host proteins to facilitate replication and to overcome host antiviral mechanisms. So he combines molecular biology, biochemistry, cell biology, and evolutionary biology to try to understand viral replication that can lead to the development of antivirals and vaccines. So I want to thank Professor Fregoso for kind of giving us some general comments, and he will also, if you have questions as he talks, please continue to put them in. So let me introduce you, Dr. Fregoso.
All right, hello, how is everybody? Well, I guess I probably can't hear you, but I need to apologize. My dog just started to bark. She was quiet through all of Jamie's presentation. She was really excited about learning everything Jamie had to say, but now she just started to bark. So hopefully she stays quiet for my talk. So thank you for that introduction, Victoria. I'm really excited to be here talking with you guys this evening. And so I'm going to tell you just a few little interesting tidbits about coronaviruses, some things that I find really fascinating about coronaviruses. So I teach our undergrad virology course, and we actually talked a lot about coronaviruses this year for apparent reasons. And it was actually really fun because the students had tons of great questions, and I'm learning a ton about this virus as we go along. I'll tell you a little bit about where the virus came from, how it replicates, and then maybe a few thoughts on vaccines and antiviral therapies. I should say again, so as Victoria mentioned, I'm a molecular biologist and I study HIV. And so I guess many would call me a molecular virologist. And so what that really means is that I think about how a virus replicates, how a virus gets into a cell, how a virus makes more of itself, how a virus spreads to a new cell, and how it's able to avoid our antiviral mechanisms. And these have been really important for understanding not just how a virus works, but for developing therapies against these viruses, right? And as Victoria mentioned, HIV being one of those classic ones where we have really great drugs against HIV because of the molecular biology we understand about this virus. So I'll start by telling you where coronaviruses or this new SARS-CoV-2 coronavirus came from. So there's actually 7 coronaviruses that are known to be infecting humans. 4 of them are endemic, meaning that they're kind of constantly circulating, and they cause what's thought to be about 10 to 12% of common cold symptoms. So every once in a while, 10% of the colds out there are actually caused by an endemic coronavirus. Within the past 20 years, there have been 3 novel coronaviruses that have jumped species. So Jamie mentioned zoonosis or cross-species transmission. This is the idea of a virus jumping from one species to another, particularly, of course, jumping into humans. And so that was first SARS, SARS-1 as I'll often refer to it as, which happened in 2002 and stayed until about 2004 was probably about the last time that we saw SARS. MERS is another coronavirus which has jumped into humans, first identified in 2013 and is actually still currently jumping into humans to this day, but it's actually an interesting virus because it doesn't seem to replicate in humans nearly as well as either SARS or SARS-CoV-2. And then, of course, the third coronavirus or the third novel coronavirus to be infecting humans is SARS-CoV-2, which is the causative agent of the disease COVID-19. So where do these viruses come from, right? So the thought is that these viruses actually reside in bats. So the reservoir for these viruses is bats. And the idea is that they actually can live in bats and bats don't get sick from them. So what bats end up becoming are these great hosts that can actually keep a virus, allow that virus to replicate, not get sick from it, and of course, because they're flying, can fly large distances and spread a virus around. Two of these coronaviruses, so SARS-1 and MERS, have had an intermediate host, meaning that they went from bats to another species. We don't know if SARS-CoV-2 has an intermediate host yet, but what's important about those intermediate hosts and the way we think about them is they kind of prime the virus to be ready to infect humans. So now they've infected something that makes it better equipped to infect humans. This was definitely the case for HIV, where prior to jumping into humans, it actually moved into chimpanzees, and that virus got primed in chimpanzees to be able to more effectively infect humans. And so that's what's really important about these intermediate hosts. So in terms of bats being this reservoir, there's actually known 500 other coronaviruses that infect bats. And it's estimated that bats could harbor a couple thousand coronaviruses. So while in the past 20 years, we've had 3 novel coronaviruses jumping over, the chances of this continuing to happen throughout our future are quite high. And this is why I think it's incredibly important that we really try and understand to the best of our ability how these viruses work. So I'll tell you a few tidbits about these coronaviruses that I think are super interesting. And again, I'm not a coronavirus biologist. I've been learning about them. And as Jamie mentioned, kind of becoming experts because of these viruses being in the news and being so important for human health. But I do find them quite fascinating. So one is how the virus actually gets into the cell. So coronaviruses are what are called RNA viruses, meaning that the genome or the information of that virus is RNA. And coronaviruses actually have some of the largest RNA genomes. But the way that the virus actually gets into a cell is that it has a protein on its surface, which is called the spike protein. And that's what gives it this kind of corona-like, crown-like image when you look at it under a microscope. So what happens is that that spike protein will bind to a host receptor called ACE2. And then there's a secondary receptor or secondary protein in the host that's needed in order to facilitate actually the binding, not only the binding, but then the entry into that new cell. And that's called TMPRSS2. And I had to write that down because I can't remember that one. So what this means is that there's a lot of specificity to the type of cell that the virus can infect and the host that the virus infect, right? Because if you think about that, that's like a lock and key. The virus has to be able to fit the ACE2 receptor properly, and it has to be able to utilize TMPRSS2 properly in order to get into the cell. But what's really interesting about this novel coronavirus is it seems that there's actually an insertion in the spike protein that allows it to not only utilize the spike protein and TMPRSS2, but a number of other host proteins instead of TMPRSS2. And what this would suggest is that potentially this novel coronavirus, SARS-CoV-2, has an enhanced capacity for spread because of its ability to infect many different cell types. So that's one aspect of this novel coronavirus that I think is super fascinating. Another is how it makes more of its genome. So again, I mentioned that these are RNA viruses. And the way that so when a virus gets in a cell, right, what it has to do is it has to make more of itself because it can make more of its genome, has to make more viral proteins, package into a new virion and spread to a new cell. And so the way that the viral, or the enzyme that's important for making more of the genomes is something called polymerase. What polymerase will do is it will bind to the genome that's brought in with the virus, right? It'll bind that genome and it'll just processively run down that genome and make more of itself, right? Most of them just kind of bind and just like that, just run down a track, right? But there are some viruses, coronaviruses being some of them, where that polymerase actually isn't processive. It doesn't just run down a track. It actually has the ability to jump from one part of the genome to another. right? That's really important for the way that coronaviruses make different aspects of their genome. But the consequence of that is that these coronaviruses actually might have the capacity to recombine, meaning that if you have one coronavirus and another coronavirus into a cell, as the polymerase is running along and it jumps, if it jumps off of one genome and onto another genome, it can recombine and create a new one. Or you can get things like deletions or insertions, like we talked about for that spike protein that allowed for this novel, or for this expanded range of its cell infectivity, where now all of a sudden that virus maybe has new capacities. Right? And so we think that this is one of the ways that coronaviruses do such a good job of spreading from one host to another, because they have this higher capacity for mutation than some other RNA viruses. Now, I know one of the concerns that a lot of people have is, does that mean that once a coronavirus is inside a person, it's going to mutate a bunch throughout, society, and then you'll be able to, you'll be able to be infected by a new coronavirus. We don't think that's normally going to be the case, or that it's going to be able to kind of overcome the vaccine that we eventually develop. If we look at flu as an example, so flu is another virus that has a very similar mutation rate to coronaviruses. And flu takes about 2 to 3 years for it to actually be able to evolve away from the vaccine that we have against it. So in thought process, it would be coronaviruses. If they are able to mutate away from the vaccine, it'll take about 2 years. So just like you get for a flu vaccine, and I'm hopeful that you're all getting your yearly flu vaccine, chances are that if we, when the time comes that we actually develop a coronavirus vaccine, it would be something that you potentially be getting every couple of years, maybe every 2 or 3 years, as opposed to something that other vaccines where you get them once when you're young, and that presents for lifelong immunity. And so with that, I'll just finish up on kind of those 2 points. So one being the vaccine. And so just to make sure that we're all on the same level playing field here, right? The idea of a vaccine is that, am I going too long, Victoria? Okay. The idea with a vaccine is that you've told your body what this pathogen looks like, so your body's ready to see it when it actually comes in. I've been incredibly impressed with the speed at which we've been developing vaccines, or we've gotten into clinical trials for SARS-CoV-2, and I'm actually quite hopeful that we will be able to develop vaccines against this virus. And in terms of drug treatments, I'm also really impressed with the way that my colleagues have been screening drugs that are already in use for their capacity to inhibit coronaviruses. And again, I think with the continued investment of scientists and the public in really hoping to push this virus to elimination, we're actually going to be able to come up with a cure for this virus in the near future. So I'll finish with that. Thank you very much.
Thank you. That was very, very helpful. I know there's a big round of applause that none of us can hear. Several good questions that I want to bring to you, Oliver, and maybe in particular. First of all, the practical question that came in early was, when will antibody testing be available for those of us who would like it, who likely had it and are symptomatic, but we're not allowed to be tested early on?
Yeah, I think that's a great question, and it's one that I'm asking as well. I know that there's capacity in labs to do that antibody testing. There's still a little bit of a high false discovery rate, so sometimes people testing positive when maybe they should have been negative. But I think that we're not far away from that, and I'm hopeful that the healthcare system will be able to interface with the labs in order to do that. I will say that I have a colleague who's actually going into clinical trials for basically a pregnancy-like test, so a stick that will tell you with a line will tell you if you test positive for that. So hopefully that will be something that in the not too distant future we'd actually even be able to do at home.
So based on your experience with other coronaviruses, do you consider it likely, and with what degree of confidence, that victims who have recovered and have antibodies will be immune and their antibodies useful for therapeutic purposes? And
yeah, that's a great question.
You may know, and Jamie, if you have any insight, but Oliver, go first. And
yeah, Jamie, if you want to jump in, that would be great as well. I think that's a great question, and one that we're still trying to understand. And so what we know from SARS-1, the first SARS, is that we do produce a neutralizing antibody, so an antibody that will actually block infection. What's interesting is that neutralizing antibody lasts for about 2 to 3 years. And so the thought is that you should be protected against infection for kind of the immediate future. But something that actually I'm going to help out that's being done at UCLA, and I'm going to lend my kind of expertise in making viruses, is that we're actually going to start testing serum from patients who have been infected to identify what percentage of people actually have neutralizing antibodies in their serum. So whether or not that can be used as a therapeutic.
So people often ask the related question. So based on what you just said, so if you get the virus, then maybe you won't get it for at least another 2 years? Can we be certain of that at this point?
We can't be certain of that yet. We need a couple years to understand that is the difficulty. And I think we also need more testing. So the biggest thing that I would say we need is more testing. Because if you can test somebody and know that they were absolutely infected because you see a virus there, and then you can see that they had an antibody response, and then sometime later come back and test and make sure that they're not getting infected or that they were reinfected. But the hope is that we will be protected. The key there is that we need enough people actually infected in order to make us be able to kind of have enough people to be able to be to go back out in the workforce and kind of revamp everything. So
would herd immunity be more effective than vaccines?
So that is the key there. So for herd immunity to work in that context, you probably need somewhere in the order of 60 to 70% of the population to be infected. So first of all, we need more testing so that we can know what our numbers look like. But it's looking like, for example, there was a report that came out from Germany, where one region of Germany that was hit quite hard, they found about 15 to 20% of their population was actually infected. So we'd actually need potentially more people infected in order for herd immunity in order to take off at a level that would be better or as helpful as a vaccine right now.
Have any mutations of the COVID-19 surface antigens been detected that would permit reinfection of someone who already has antibodies?
Right, so
We're putting you on the spot with the hard questions, Oliver.
No, these are great questions. I love them. And what's great is that science is being done right now at an unprecedented rate for this virus. I'm seeing new papers come out daily that are just blowing my mind. And so I just saw a paper come out today that basically took patients isolated SARS-CoV-2, so they took viruses from patients. They identified 11 different versions of the virus, so they had some sort of mutation in it. 6 of those had mutations in the spike protein, meaning that 6 of those potentially had mutations that would allow them to bind to ACE2 receptor differently. They tested them in vitro, and that's the key that I want to point out. This was tested in cell culture. So basically, you put cells in a dish, and these were actually monkey cells. So you put cells in a dish, you throw the virus on there, and they measured the ability of that virus to replicate. And they found that those viruses actually had different ability to replicate in those monkey cells. So that says that there is an inherent capacity for the virus to replicate a little bit differently based on those mutations, but it's going to be a very different picture when you consider both the innate and adaptive immune response of a person. And so I would say the answer to that question is no, not yet. Those mutations have not been identified. And again, thinking about flu, it takes about 2 years for flu to evolve to the case where now it's basically overcome the vaccine that we currently have.
Do you happen to know how the trial of the Gilead drug remdesivir is doing?
I wish I had inside knowledge of that. I'm really curious. So there's one of the molecular mechanisms of the virus that I was thinking about talking about is again, this idea of polymerase, so making more of the genome. So coronaviruses are really interesting in that context because they actually have the ability to proofread. They have the ability to say, you know what, I've put a base here that's actually the incorrect base. And so I'm going to take that base out and I'm going to put in the correct base. Most viruses cannot do that, but coronaviruses can. Remdesivir, the way that it works is it actually blocks up the ability of that polymerase to work because it terminates that polymerase from moving on. So I'm really curious to know if the ability of the viral polymerase to actually recognize an incorrect base and pull it out and put a correct one in, if it would be able to overcome remdesivir, because that's kind of what remdesivir is doing, is putting a bad base in there to stop polymerase from acting so the virus doesn't make any more of the genome. So I've heard that it's working well because you basically overcome the virus. You have too much of that in there, but I'm really curious to see how it goes. So I have high hopes for remdesivir.
I'll ask one more question, then I'm going to move on to our speaker. And we have many more questions I didn't get to. I may get to them at the end. But there's so much variability in the severity of infections. What are the likely explanations? How important is the initial viral load likely to be?
Yeah, another really good question. And so people are identifying comorbidities or factors in people's lives that actually contribute to the severity of the disease. And there are definitely a handful of those. The viral load was one that was considered originally, and that's one that's still really unclear. And so for those who don't know, the idea of viral load is how much virus actually initially gets in, or how much virus actually is in the patient. What we do know is that the severity of the disease comes from a cytokine storm, which is basically what happens there is that the virus actually blocks your initial response to the virus. And so that allows the virus to make more of itself. And then eventually, you're going to have this innate immune response that's going to kick on. And when that kicks on, it kicks on at a point that's way too late. And so you make a ton of these cytokines. And what they do is then they recruit these cells that their job is to come in and eat cells that are infected to get rid of the dead infected cells. But now they've come in, and what they're actually doing is they're injuring the lung because there's so many of them coming in and swarming. And so what leads to that is still quite unclear in terms of why some people have a severe cytokine storm and others do not.
So thank you, Oliver. So both Professors Fregoso and Lloyd-Smith, they're going to hang on because we may have questions at the end that they can address. But I want to introduce our last panelist, and this is Professor Julian Bauer. She received her PhD in psychology from UCLA with a focus on clinical and health psychology. She's currently a professor in that department and also in the Psychiatry, Biobehavioral Sciences at UCLA. And she holds a George F. Salon Chair for Psychobiology in the Cousins Center for Psychoneuroimmunology at UCLA. And she's president of the Psychoneuroimmunology Research Society. So we want to know what psychoneuroimmunology, she's going to be talking to give you some aspect of that. But she focuses on the bidirectional interactions between the psychological behavioral states and the immune system among individuals facing stressful life events, particularly cancer patients and survivors. So she's particularly interested in risk and resilience factors that can influence those neuroimmune interactions and the use of mind-body interventions to enhance health and well-being. So including mindfulness meditation. So I think this is relevant about why some people can really be severely affected. And with that, I hope that Professor Bauer will put on her video and her speaker.
That I should be here. Well, thank you so much, Dean Sork. It's a pleasure to be here to join this, my amazing colleagues. I've already learned a lot about the coronavirus. So as Dean Sork indicated, I am a psychologist with expertise in psychoneuroimmunology. And I think the last question was a great kind of prelude for my talk, because really all the biology that we've just heard about takes place inside of a person. And immunologists tend to look, I think, somewhat narrowly at the dynamic interactions between viruses and immune cells. But as psychologists and psychoneuroimmunologists, we're particularly interested in how those interactions are actually shaped by the host and by psychological processes occurring in the host. So I'm particularly interested in stressful life events, how those affect the immune system, and what we can do to buffer those effects. So I think at this point, we kind of all have this basic assumption, a basic understanding that stress is relevant for the immune system. But I think it's important to just remember that really the rigorous science in this area didn't emerge until the 1980s, where some really elegant work was done showing that stressors ranging from going through medical school exams to taking care of a loved one with Alzheimer's dementia those kind of stressful life events were related to decrements in the immune response, including decreases in antiviral immunity. And this work has now, expanded considerably over the last 10, 20, 30 years to show that a range of different stressful events can influence the immune system. And importantly, this work has also really beautifully documented the very close connections between the brain and the immune system. And so we know how intimately these systems are interconnected, such that stress can regulate what kinds of immune cells are made by the bone marrow, where they go in the body, and how they function when they get there. So one of the stressors I really wanted to highlight today is social stress, because it turns out that threats to our social self turn out to be very potent in terms of our immune response. So this includes stressors like feeling left out or socially rejected, can include things like low social status, but also include feeling socially isolated or lonely. And this is, I think, particularly relevant for antiviral immunity, because there's actually a growing body of research showing that the experience of social isolation actually skews the immune system in a way that makes it less adept at dealing with viruses. And this has been shown with viruses like the cold, but also in the context of HIV. So this question of, why do some people seem to develop symptoms and get sick and die and others don't with in the context of COVID-19, this was a big question in the context of the HIV epidemic. Why are some people dying so quickly and other people are living a long time? And one factor that may be relevant in that context and potentially in this one is this experience of social isolation. So as Victoria said, one of the key things my research focuses on is, so how do we counter this? How do we, in the absence of having perhaps medications that can fight these viruses in our bodies, are there any psychological approaches we can use that can help bolster our antiviral immunity? So work done in my lab in collaborations with colleagues at UCLA and elsewhere has identified a number of different factors, and I'll highlight two of them today. One is pretty straightforward, which is sleep. Sleep is a critical modulator of the immune system. And for me, based on all the research I've seen showing how important sleep is for developing a healthy immune system, when I'm trying to make decisions between things like physical activity and sleep, I tend to prioritize sleep because it really is critical for, brain, body, and immune system health. The other factor I want to highlight, which you may have heard less about, is the importance of positive emotions for our immune systems. So in psychoneuroimmunology and psychology in general, we tend to focus a lot on stress and anxiety and depression and the negative effects of these things on our immune system. But there's a growing body of science showing that positive psychological states may be just as important, actually, if not more important, for developing a healthy immune system. So what our group and others have shown is that people who experience more happiness, more joy, who have a deeper sense of meaning and purpose in life, seem to have a healthier immune system, including stronger antiviral immunity. So we're very interested in what approaches or techniques we can all use to be helping to cultivate this healthy immune system. And one approach we've been particularly interested in is mindfulness meditation. So there's a lot of hype around mindfulness meditation, and in some cases, I think the ideas that the public has about mindfulness go beyond the data. But there's actually very solid empirical basis that mindfulness meditation programs can be beneficial for our mental and potentially for our physical and immunological health. So we have been testing a 6-week mindfulness program that was developed here at UCLA in our Mindful Awareness Research Center. And we've done this with highly stressed populations, including breast cancer patients and survivors. And our work has shown that mindfulness, even this pretty short intervention, 6 weeks, once a week, leads to decreases in depression and stress, decreases in loneliness, increases in sleep quality, increases in positive psychological states that in many cases are actually more enduring than the effects on stress and depression. And importantly, we've shown that mindfulness can shift the immune system in a way that should bolster antiviral immunity and decrease pro-inflammatory cytokines, which are contributing to this cytokine storm that we just heard about. So I think what this research suggests is that even in the context of very severe situations, what we've been studying again is people dealing with a breast cancer diagnosis, but perhaps in the context of the pandemic we're all experiencing now, there are evidence-based strategies we can use to enhance not only our psychological well-being, but perhaps our immune well-being as well. And I should just note that this mindfulness program is actually available. You can get on to the UCLA website and enroll either in the 6-week course, or they even have just these little bites of mindfulness where you spend 5 minutes kind of cultivating a mindful state of being, sending loving kindness to others and yourself. And again, our research suggests that that may actually be beneficial for our physical health and immunity.
Wonderful. That was great to hear some comments about positive things can do. So there's one question for you, Julie, which is when you think about the fact that we're seeing disproportionate infection to both low-income individuals of various ethnic and racial categories. How much do you think it's the stress? First of all, a lot of those people are working and perhaps more exposed, but perhaps they're also working and exposed under conditions creating higher stress and therefore making it more likely. Do you want to comment on the interaction between the people who are getting this disease and?
So, there is a well-known link between socioeconomic status and physical health. And that's something psychologists have been very interested in because there's so many factors, as you're noting, Victoria, that could be playing into those kind of associations. But there's pretty compelling evidence that psychological stress may be playing a role, and that the stress associated with having, both potentially experiencing more stressors and having fewer resources to help manage those stressors may be part of the reason why you're seeing kind of particularly pronounced effects in certain populations. Another pathway we should talk about here, though, is health behaviors, because another thing that may be playing a role in these associations is that stress can diminish your ability to sleep, exercise, eat well, leading to metabolic diseases, which we know may be comorbidities that are helping to fuel increased prevalence rates in certain populations.
Great. So what I think I'd like to do is ask the three panelists all to rejoin the conversation, turn on your video, perhaps unmute your phones. And we have a few minutes left, so I'm going to actually bring up some more general questions. And one question that is kind of general, but I could imagine more than one answer. So, okay, is this pandemic a once-in-a-lifetime event, or can we expect another novel virus to affect the world this way? Would we be better prepared for another novel virus in the future? So we have two panelists with evolutionary perspectives. Do each of you want to give your comments on this?
I'll jump in dangerously. We can hope that this pandemic in its scale and impact is a once in a lifetime event, but it's certainly not a guarantee. There's been a lot of comparison to the 1918 so-called Spanish influenza pandemic, which was the last one to really rock the world to this degree. It happens to be about 100 years ago. There is no clock that says these things happen every 100 years. I would say that, there's been a lot of science done and a lot of awareness that as humans, we're doing a lot of things which increase the risk of new viruses spilling into the human population. In particular, cutting into a lot of ecosystems that previously had been very remote, having a lot more contact with a lot more different kinds of animals at large scales than we did previously. And just the increasing human mobility, it's entirely likely that perhaps a virus like this one, certainly, ancestral versions of HIV, viruses like that have probably been spilling over into human populations for millennia, but not getting very far because populations weren't big enough, people weren't moving around enough to enable a full pandemic to occur. In the modern era, mobility is increasing exponentially, and so viruses can easily get around the world, in hours. And that is a changed playing field. I guess one last thing I would point out is that while this virus has really gotten everyone's attention to a very high degree, it's hardly the first virus to make global impacts in recent times. Like HIV was a tremendous pandemic of a very different nature. But even in recent years, we've seen Zika virus basically jumping between hemispheres and having huge impacts. We've seen dengue and chikungunya viruses, which are other mosquito-borne viruses that are really increasing alarmingly. And so unfortunately, I think, if you look at the pattern, we should expect there to be more sort of incursions, more viruses testing the boundaries. How far they get is a big question. Whether we'll be better prepared is an excellent question. And I think much of that comes down to how much we're able to learn as a society, some of the lessons from this one. I think in some ways we've done better than we did with some past pandemics, but there've certainly been huge errors made. And I'll just say that, politicians have been all over the map in terms of how they address this crisis and the degree to which they're relying on the science, which is actually, as Oliver said, moving incredibly fast, I think, on this. Anyway, I'll let the rest of the panel weigh in.
Oliver, do you have anything to add in your predictions?
No, I mean, Jamie basically said it all. I mean, I agree. I think the one thing, I guess maybe the one thing that I'll add is that, one aspect of this virus is how well it actually spreads between people. And in addition, the data coming out suggesting or indicating that actually peak viral loads, you're producing the most amount of virus 2 days before you actually become sick. And so that's another reason. So this virus, in a way, is kind of unique, right? If you think about the first SARS and MERS, those haven't gone pandemic. So it's a combination of what Jamie said about us encroaching on landscapes, and I could talk for hours about HIV and why that became a pandemic, and the fact that those viruses have been jumping into humans for a very, very long time in the past. But there's also something about the virus that also has to fit properly to allow it to become a pandemic to this scale.
Great. Early on, we got a question that I'd like to bring forward now. Is actually while Jamie was talking, and it says, thank you for your outstanding presentation. Given your work describing the importance of viral burden, what are the implications of potential pharmacological preventative measures? And you might start, and then maybe Oliver can add to that one as well.
Yeah, that's a great question. And I think preventative measures, so I would make a distinction between, or just to be clear, it sounds like the person asking the question has a clear idea, but there's a difference between a pharmacological treatment, which is to say I get sick and I take a drug to try to combat the sickness, and a preventive measure, which is like a pill you take prophylactically because you think you may have been exposed, or maybe you take it as a generality. I'm not aware of any work on prophylactic treatment, preventative treatment for COVID, but we're still trying to figure out if any of the drugs work. the short answer is that anything you can do which knocks down the viral load will diminish the amount of virus which is shed, and will, proportionately diminish the doses that people are exposed to. And while I made a point that we don't yet understand what the infectious dose level is, which is needed to, there's a so-called ID50, which gives a 50% chance of infecting somebody. We don't know that number yet. Anything you do to reduce the dose level in exposures will reduce transmission. And here HIV again is a real touchstone, because the drugs have gotten so good that they can reduce the virus in infected people down to undetectable levels. And in the time that I've been in this field, that went from a sort of crazy idea that you could use drugs to reduce transmission and stop the pandemic, to now it's absolutely the go-to technique. So maybe we get there for COVID. I don't know, it might be hard to identify people at that particular risk who you would choose to treat prophylactically. But I could certainly imagine if we had a drug that giving it to people who are at high occupational risk, like healthcare workers, could turn out to be a viable strategy. But that's well outside my bailiwick.
Anyone else want to comment on that? That's all right. Here's a question for anyone on the panel. Many of us have read the fact that men seem to be more affected by this coronavirus. Any of you want to either speculate or perhaps knowledgeably answer why is it that men seem to be more susceptible?
I can hit the speculating side if others aren't jumping in. I'll say a few things. First of all, the pattern came out very early in the data from China. And at the time, many people attributed it to the fact that men in China smoke at very high levels. And smoking is known to be an aggravating factor and to make you more susceptible to infection and disease. The pattern has continued to hold across other societies where probably the disparity in smoking levels isn't as strong. And I honestly don't know, because I've not been able to keep up with all of the studies that are flooding the literature, whether people have done proper sort of controlled epidemiological analyses that account for all the other comorbidities that come with being male, like higher levels of cardiovascular disease, possible smoking, and so on, and seeing whether this pattern still holds. I will say that it's a relatively common pattern in infectious disease across the animal kingdom, that males are often more heavily affected. And some of this, I expect Julian Oliver may be more knowledgeable about this than I am, but some of it is thought to have to do with different hormonal states in men and women, and how those, like testosterone, impacts the immune system function.
Any other comments from? So
I'll just point out that the countries that have done the best at stopping or slowing the progression have been largely led by women. So other side of the argument there, but yes.
Thanks for that useful fact.
It's a striking pattern.
Yeah.
It is.
Yeah. So Professor Bauer, what do you measure to assess a healthy immune system?
Yeah, great question. I was using that term very kind of globally, but there's a number of different ways we can assess immune system function. I think in this, one of the ways that we, it depends, I guess my answer is, it depends on what aspect of the immune system you're interested in looking at. So increasingly in my field, we tend to look at levels of inflammatory cytokines and the genes that regulate pro-inflammatory cytokines, because we know how important inflammation is for many of the chronic diseases that we study, like cardiovascular disease and cancer, but are also relevant in the context of HIV and viral infection. So that's kind of a go-to immune outcome that we're trying to reduce. But we also, another important aspect of the immune system is the ability to fight against viruses. So we will also look at the ability of the immune system to make antibodies in response to vaccination, or even just expression of antiviral genes in immune cells. So we will look at it at multiple levels. So what the immune cells are doing, what kind of genes are getting expressed in different immune cells, and how good they are at making antibodies, et cetera.
I'm not sure who wants to answer this one, but you can all sort of, you're all listening to the experts, as do I. Are the experts certain that social distancing, which I will parenthetically say really means physical distancing? In fact, I'm going to go off on a tangent because there's been some pointing that social distancing means isolation, and isolation does lead to stress. And so one of the things to counteract during this period of physical distancing is not to have social distancing. But with my parenthetical comment aside, if we do physical distancing, are we really certain that's going to reduce the causes of mortality, reduce it over the long term? And anyone want to just comment on that question?
I mean, I think one of the aspects of this that we haven't really talked about is the healthcare system, right? And so part of the reason why it was so important to flatten the curve, as the term that we all like to use, was so that we didn't overburden our healthcare system to the extent that we would not be able to handle this. And that has, the data suggests that worked. And so that's not asking, that's not answering the question about if it's going to prevent people from getting infected, right? Because we're still susceptible. That is the bottom line, right? But I think the important aspect of the physical distancing, or distantly socializing, as I've been told to also call it, is that it also really helps to just slow the progression so that our health systems would not be overburdened and people wouldn't die because they couldn't actually be helped.
Okay, well, listen, our time is up. We've got questions we couldn't get to, and to those that raised them, I apologize, but the panelists I really appreciate all of your comments, and if we could, we'd all clap and thank you, Professor Jamie Lloyd-Smith, Professor Oliver Fregoso, and Professor Julie Bauer for coming and commenting on some of the very few elements and components of this disease. I want to thank all of the people who came, because I know you, and I just wish I was there to go around and say thank you for coming and have a conversation and ask how all you are doing. But I do really care about you and hope that you're doing well, staying healthy, and everyone around you is staying healthy. So with that, thank you for participating in this wonderful, hopefully not too common occurrence of our virtual salon. And again, I really appreciate it. And thank you to all of the staff that made sure that the logistics of this event came on. So thank you very much, and good night, and be healthy.
Good night.